Good afternoon. My name is Narita Thungsatianpan. I am a master degree student from Cellular University. I would like to present my research in the topic of cell penetrating peptide nanocomplex enhance cellular uptake of the dsRNA in SF9 cell line. RNA interference or RNAs have been applied to study the functions of genes in a wide range of organisms. The process involves the interference of the expressions of homologous genes after transcription by specific dsRNA. The mechanism of RNAi is shown in the right panel. Long duplex RNA is cleaved into short duplex strand with 20 to 25 nucleotide by dicer. Then agonal protein lead to unwired siRNA and consequently incorporated into this complex. Sense strand is removed while antisense strand remain bound with the complex. Due to this position, siRNA can match with mRNA target in version and quick manner. Finally, mRNA is degraded. Currently, growing number of research groups are exploring the efficacy of using the dsRNA in agriculture. Among RNAi applications, therapeutic silencing of disease-causing genes has received attention to developing viral vaccine in shrimp. Furthermore, RNAi technology are being developed to apply in insect pest control. Additionally, insect cell line has used as a model for developing RNAi in arthropods. However, premature degradation of the dsRNA after ingestion and poor cellular uptake currently limit this approach for pest management. As we focus on processing and cell internalizations of the dsRNA in insects, there are different RNAi in Coleoptera and Lepidoptera. In Coleoptera, the dsRNA is released from the endosomes and processed by the core RNAi machinery. While in Lepidoptera, the dsRNA is not released from the endosomes, avoiding the dsRNA contact with the core RNAi machinery. Likewise, from the previous study of insects, two mechanisms of the dsRNA cellular uptake have been identified, including cyst-like transmembrane channel and endocytosis. As mentioned above, multiple mechanisms have been affect the RNAi efficiency in different insect species. The next barrier is cellular uptake of the dsRNA due to their shape and size. Additionally, nuclease inside hemolymph and midgut appear to affect the stability of the dsRNA. Then, many efforts have been made to overcome this physiological and cellular uptake barrier. Another way to overcome is using nanocarrier that could be increase the stability of the dsRNA in the insect body or increase cellular uptake of the dsRNA. During RNAi through feeding, nanoparticle can act as molecular carrier that could promote the translocation of the dsRNA across the epithelial cell. Two proposed mechanisms include direct fusion with cell membrane and endocytosis. Some example of nanoparticle-based delivery system that have been used in insects are chitosan and branch amphiphilic peptide capsule. In this study, we are interested in using cell penetrating peptide or CPP as the dsRNA delivery, the dsRNA carrier. CPP are cationic peptide composing of 8 to 30 amino acids, which can bind to any cell surface and easily deliver molecules across cell membrane. It became a popular tool for delivery of various cargo, including peptide, DNA, and drug. And the CPP internalization mechanisms appear into two classes, include non-energy or energy-dependent pathway. In objective, we explore the possibility of using CPP as a carrier to improve cellular uptake of long-duration RNA in arthropod cell line. And the peptide sequence is shown in this table. Outline of the experiment. Ratio between CPP and the duration RNA for nanocomplex formation were characterized. Next, the physical property and morphology of nanocomplex were characterized, and then cellular uptake efficiency of the duration RNA were compared between two CPP in SF9. For expressions and isolation of the duration RNA, E. coli containing plasmid with RR2 gene were cultured and induced by IPTG. Then cell platelets were suspended with 0.1% SDS, and single-strand RNA was eliminated by RNase. The duration RNA extraction was performed according to manufacturer instructions of style reagent. The quality of extract the duration RNA was evaluated via spectrophotometry and gel electrophoresis. The results suggest that size of extract the duration RNA was approximately to 600 base pair. That's corresponding to the expected size of RR2 gene. After the duration RNA production, various concentration of peptide were mixed with 1 microgram of the duration RNA in nucleate-free water at different P to N ratio, where P is number of positive charge and N is number of negative charge. Then the mixture were incubated at room temperature for 30 minutes. Next, gel retardation assay was performed. Moreover, the complexation between the duration RNA and CPP were prepared by varying positive to negative charge ratio, ranging from 0.5 to 5. After 30 minutes of incubation, 0.1 ml of quantity-full RNA dye were added into each well. The fluorescent intensity were measured using micropage reader. Here, negatively charged of the duration RNA was neutralized by the positive charge of CPP, resulting in retardation the mobility of the duration RNA. At 0.1 to 1 ratio, band of the duration RNA was obviously seen, and gradually shifted into higher molecular weight, whereas at 2 to 1 ratio, no duration RNA band was observed. Due to the peptide was able to bind the duration RNA and completely retarded the mobility of the duration RNA. This similar trend was observed in higher positive to negative charge ratio. In quantitative fluorescent intensity assay, the quenching of RNA by inflorescent dye was considered as an indicator for complexation between the duration RNA and peptide. The results show that mean fluorescent intensity was continuously decreased with the increasing of positive to negative charge ratio. This trend was observed in nanocomplex prepared with TAT and EB1 peptide. Then the quantitative experiments is corresponding well to the experiment from gel retardation assay. Next, physical property of the nanocomplex were characterized by the LS technique. Briefly, various concentration of CPP were mixed with the duration RNA, and the mixture were agitated by pipetting and vortexing, and the mixture were incubated at room temperature for 30 minutes. Next, Zeta potential and size were measured using dynamic bright scattering technique. In addition, transmission electron microscopy was performed on the complex at 50:1 ratio of both peptides. Briefly, the solution was dropped onto the copper kit and allowed to dry for 30 minutes. Then the mixture was stained with 2% uranium acetate and analyzed under 120 kilovolts. As you can see, hydrodynamic size of 0.121 of the nanocomplex prepared with TAT and double-strand RNA had the largest particle size with the polydispersity intensity index close to 1, implying a broad range of size distribution. And after increasing the positive to negative charge ratio, size was decreased around to 200 nanometers with a low PDI, indicating that a homogeneous size distributions. Likewise, in zeta potential, upon increasing the amount of the CPP, zeta potential increased as expected to 22.9 and 25.7 millivolt for TAT and EB1 nanocomplex respectively. So positive to negative charge ratio of 50:1 had the constant hydrodynamic size with a low PDI, and the maximum positively charged was also presented at this ratio. Then 50:1 ratio were selected for further evaluation for size distributions and morphology by TEM. In the TEM image show the morphology of TAT and double-strand RNA nanocomplex was spherical shape with the average size of 29 nanometer, while the morphology of EB1 and double-strand RNA appear in spherical shape with the average size of 17 nanometer. Based on their hydrodynamic size, PDI value, and zeta potential, the nanocomplex at 50:1 and 1:1 positive charge to negative charge ratio were used to analyze the cellular uptake efficiency of the double-strand RNA. SF9 were set on 24-well plate for at 24-well plate and culture for 18 hours. Next, cells were incubated with the nanocomplex. Cells treated with 1 microgram of the double-strand RNA were used as a control, and all conditions were performed in triplicate. After 6 and 48 hours of incubations, cells were washed with PBS for remove uninternalized double-strand RNA and added by reagent for extract total RNA. Next, 2 micrograms of RNA were synthesized to the first-strand cDNA, and finally, semi-quantitative PCR was performed and subjected to image for analyze cellular uptake analysis. The results show that in these results, RR2 band intensity was considered as cellular uptake efficiency of the double-strand RNA. In the absence of CPP complexing, no double-strand RNA was observed. Events after 48 hours of incubation and at 1:1 positive to negative charge ratio, double-strand RNA was not significantly internalization into cells due to their negative charge character as expected. Whereas at 50:1 ratio, double-strand RNA were internalized into cell as early as 1 hour and remain after 6 hours of incubation. The results suggest that both of TAT and EB1 were able to carry 600 base pair of double-strand RNA across the cell membrane into cell cytoplasm. despite not significantly different at 1 hour. Double-strand RNA delivered into cells by EB1 remain detectable after 48 hours. In contrast, double-strand RNA level was obviously diminished in TAT-mediated delivery. For discussions, the distinct double-strand RNA level after prolonged incubation periods could be influenced by the sequence of CPP. Task force endosomal entrapment of which cargo were trapped inside the endosome and lysosome. While in EB1, it is a improved CPP for endosomal escape. The sequence of EB1 contains histidine and tryptophan that induce endosomal bursting. As viral mRNA targets for racking in the SF9 cell line, remaining double-strand RNA after prolonged incubation was consequently available as a template for PCR amplifications. In conclusions, double-strand RNA bind non-covalently to cationic CPP and at an appropriate ratio, cationic complex which size in nanometer range can be formed. The physical properties such as zeta potential and size depend on the charge ratio that govern the internalization efficiency into cells. And finally, I would like to thank Assistant Professor Chomda Sinthuvanit, Associate Professor Sasimanat, and Associate Professor Nathanan for advice and support me throughout the research. And I also thank Dr. Lapipat Maruchak for plasmid containing RRUgene. And the research is supported by graduate scholarship from Graduate School, Kasetsat University, and Kasetsat University Research and Department Institute Ghent. Thank you for your attention.
Okay, thank you very much, Ms. Narita, for nice presentation. And are there any questions from the audience? Okay, I have some questions. I would like to know about the mechanism, how to form nanocomplex between CPP and 600 base pair double-strand RNA. Do you know about the mechanism to form nanoparticle complex?
And based on their chart, double-strand RNA have negative charge and CPP or peptide have positive charge. So when we mix them together, it can bind cell assembly through electrostatic interaction.
And at the why did the double-strand RNA level decrease with TAT-mediated delivery? For example, at the 48 hours when compared with the EB1.
Because as we focus on the primary sequence of TAT, and previous studies suggest that TAT can occur the endosomal entrapment and consequent to the lysosome for degradations. And I think, and I guess, because this point, double-strand RNA level mediated by TAT diminish after 48 hours because it's subjected to lysosome, while in the EB1, it have histidines that can promote the proton scavenger. So it can escape from the endosome and release the double-strand RNA in the cytosol. So we can detect the double-strand RNA after incubation at 48 hours.
Okay. From your result, what did you plan to do in the further study? Your idea?
Maybe study about the mechanisms of EB1 after prolonged incubation.
Okay. Are there any questions? Okay. Thank you very much. thank you very much to the audience for your attending in this section.